Where the connections are.
Yeah.
Thanks for coming. Thanks for attending our workshop and from all around the world. I know that like it's very early in the morning for some of our attendee and late at night for some others. So thank you so much. Really appreciate that you are interested and participating in our workshop. I'm Yasaman Gholami. You can call me Yasmin. I'm a fourth-year PhD student in architecture at Georgia Tech under supervision of Dr. Samin Bafna. My focus is on healthcare and human-computer interaction, specifically extended reality. And I'm also a graduate research assistant with a shared appointment at Integrated Design Lab with the direction of Dr. Mihai and at Georgia Tech Research Institute with the direction of Dr. Paula Gomez. And I'm here with my amazing colleague, Simin Nasiri. She has a very multidisciplinary background, both professionally and academically, in art, computational design, architecture, and now brain and cognitive science. So do you want to take over?
Sure. Hi, everybody, and thank you very much for coming today. We're truly honored for having you here. And our online people, that I saw that they were super excited because they are talking about how diverse it is from India, Iran, Turkey, Canada, US, yeah, South America, I guess, where else? There are a lot, yeah, going on, so from Europe. So we're so happy to have you all here, and that's a true honor. So as Yasaman said, my name is Simin, and I'm doing my second-year PhD in cognitive psychology and brain sciences. My background was in architecture, computational design, and my advisor is Dr. Thackeray Brown, that we're honored to have him here. So what else? I guess, yeah, what I'm doing is studying morphology and, space layouts, how that impacts our brain and behavior. So Yasaman and I could run this workshop with the help of Neuroscience Initiative, Center for Research and Education and Navigation, Crane, School of Architecture and School of Psychology. Without the help of these people, it would have been super hard for us. So the agenda or the schedule of today will be done with the opening around 9:40, which we are getting there. And then we'll have an overview of data collection methods and studying human and built environment until 10 a.m. And then we'll have our first keynote talk by Dr. Samin Bafna, and the topic of the talk will be the cognitively shaped environment, a primer on descriptive methods with the Q&A. So then we'll have our first coffee break until 11:25, and then we'll have the behavioral and physiological data collection methods by Yasaman, and then we'll have our second keynote speaker, Dr. Thackeray Brown, with the topic of brains without borders, how manipulating barriers and environment structure shapes our memory for better or worse. And the Q&A, then we'll have our lunch and social break until 2 p.m. that, so we'll have lunch here. We don't exactly know what our online people will have, but yeah, you can definitely network, talk. We'll have your breakout rooms to interact with each other there. So that will be for our online people, and then we'll continue with reviewing the schedule once again, neuroscience methods until 2:30 p.m. And then our third keynote talk by Dr. Hugo Spears on how the brain responds to spatial structure and aesthetics in the built environment with an fMRI study and a Q&A, and then interaction of demo presenters. We'll have some wonderful demonstrations of some of the devices and methods that we're introducing here until 3:50 p.m. And then we'll have our second coffee break. Then will be our second session with the interactive demo workshop. So that will be the schedule of today. And so the objective of today, our general objective is that we want to provide an overview of common neuroscience data collection methods and for built environment studies. Why we decided to do that? Yasaman and I had our separate but overlapping path of studying environment and how environment impacts humans. So we knew that that's our passion. But at some point we were like, so we need something organized to give us an introduction of what is brain, how brain works, how environment impacts brain, and all of that, and how we can study brain and environment. So that was when we were like, so we need it. So let's start something. Maybe we can hopefully have some other people just benefiting from that. So that's our main objective. Okay. Of today. And then what do we mean by neuroscience methods? So we know that neuroscience is studying of neural data, but in our literature of neuroscience and built environment, and as it's also known as neuroarchitecture, we call behavioral data, physiological data, and neural data all together neuroscience methods, which basically physiological data and neural data will be our main focus, but then behavioral data is the complementary basically studies to support the quantitative or neural data that we're collecting. So yeah, I guess I just said that. Yes, Simin?
Yeah, thank you, Simin. So I'm assuming that everyone here knows that there is an interplay between human and built environment, where humans shape the built environment and then get influenced by the environment. So why there are many disciplines that offer different approaches to studying the human built environment interaction relationship, including medicine, public health, architecture, planning, and then they have their own approaches, their own scales, from urban scales to the building scale and interior design. And then we have different, settings, either physical settings or computer mediated settings. All of these disciplines and approaches need their unique kind of data. And from the human side, there are three, let's say, type of data that can let's say there are some impacts that can be captured through patterns of behavior, and then we can capture those by behavioral data. And then there are some impacts that can be captured through body-based indicators, and we can capture those by physiological data. And there are some impacts that can be captured through neural activity, and we can capture them through neural data. And of course, some of this impact can be captured with combination of these data or all of them. And then from the built environment side, we can have data. We can have two types of data. One is raw data, which can be captured through environmental sensors or measurements. And then there are another set of data that are processed data that can be captured through a space syntax method and GIS. So the three common human side data, behavioral, physiological, and neural data, will be covered by Simin's and my presentation. And the space syntax method will be covered by talk of Dr. Sanit Bafna. And then the interplay between environment and human will be covered by talk of Dr. Thackeray Brown and Dr. Hugo Spears. And then in our final session, in our demo session, we will cover techniques on both sides, from human and built environment side, physiological and neural data, and also space syntax techniques and agent-based simulation for the built environment. We are very honored to have three of the best in our field. Dr. Sanit Bafna, he's Associate Professor in the School of Architecture at Georgia Tech. And Dr. Thackeray Brown, Associate Professor in the School of Psychology at Georgia Tech. And Dr. Hugo Spears, Professor in Cognitive Neuroscience in UCL. So for behavioral data, the common method that we're gonna discuss would be observation, survey, interview, automated data collection, and digital trace. In physiological data, we will discuss ECG, PPG, BP blood pressure, EDA or GSR, galvanic skin response, skin temperature, electromyography, EMG, and eye tracking. And in neural data, among common methods in studying neuroscience and built environment, we will cover fMRI and event-related potential, including EEG and MEG, and DTI, single unit recording and legend. And the purpose, as I mentioned before, is like to provide an organized overview of these common methods rather than getting deep into each or like provide details. So yeah, this is like the purpose of the workshop. And to begin with, Simin will present us brain structure first.
So, as we know that we have a wide range from architects to neuroscientists and everyone in between who is interested in the field. So we're going to go, as Yasaman said, very introductory from the neuro side for anyone interested. And then our guest speakers will cover more with the state-of-the-art research that they're doing. So I'll start by neuroscience methods to study environment. Why should we do that though? So we have human and environment, and we know that they have this interactive, always connection that we are not really going to go into the details, but we ask this question: which one do you think impacts the other first, or which one do you think in general impacts the others? Can we say environment shaped us, or we shape our environment? I guess we're not going to go into the arguments of anthropological or old archaeological arguments of which one started which, but we know that we have this interaction right now, and that's the most important interaction of our body, our psyche, us, our consciousness with basically our environment and our life. So that's why we focus on studying human environment interaction. And if we want to know that interaction, we want to know how we shape environment and how environments basically impacts our brain. So to study brain, we need to study brain basically for a better and healthier environment. Again, for a better and healthier us to have a better life experience. That's why we need to study brain for environment. Because I remember that I was always being asked this question of, so what do they have to do with each other? You say that your background was in architecture. What are you doing in psychology and neuroscience? I guess they are very much related. So now let's nerd out the nerves or unnerve. Oh, never mind. So anyways, so we'll start with neurons, which are the base of everything that we're going to talk about. So neurons we're going to go basically bottom up instead of going from the brain. So this is what makes everything that we are going to talk about. So these little cells consist of cell body, dendrites, axon, or nerve fibers. We are not going to go into the details, so you can definitely go into the neuroscience books and nerd out there. But here we just mentioned this to use it later. The way they talk to each other is through these ions, and how basically their electrical charge that they can transfer the information. Whatever happens, we make some transformation of those information into these ions and electrical charge that they can transfer information through the connections of nerves. So what is nervous system? We're just going to go quickly on, we have two, it consists of two main parts. We call it central nervous system, CNS, which includes brain and spinal cord, and peripheral nervous system, PNS, which consists of spinal nerves and cranial nerves. So what the peripheral nervous system does is takes the information from our brain or the central nervous system and distributes it to the other parts of our body, or takes the information from our environment and transfers that into our central nervous system. So with a focus on central nervous system today, do we think that brain have a functional architecture with respects to the components, or if it has some faculties basically independently working and being responsible for some activities, like what we remember from phrenological images? Back then, they thought that we have specific parts in our brain responsible for something like justice, I don't know, studying, something like that. But now we know that it's not exactly like that, but not that they were super unrelated. So, or the other theory is that the brain is totally a whole thing working together, and there is nothing as separate faculties or separate regions doing things. So which one is correct? Yeah, both. So they work together connectedly, but we also today know that we have specific regions that are responsible for some things more than the other. So with that, knowing that we have regions, now we can ask, where do we study for what? For example, if we are interested in studying environment and environmental factors and, basically variables, so what are we going to measure, or where are we going to study the brain? So we're just going to go quickly in the most generic way of dividing the brain by midbrain and the cortex. So midbrain will be responsible for more of our basically what is it called, survival mode. So it'll be basal ganglia, limbic system, and whatever is inside it, like thalamus, amygdala, hippocampus, and hypothalamus. So all of this together, basically our midbrain is more responsible for sensations, emotions, motor, and memory. So we are going to learn memory. We have our emotions mostly there, and we have our smell perception, plus we have our hormone regulation that we know is kind of everything, and our response to fear, fight, and flight, and sensations processing, and also our reflective reactions. And cortex is basically responsible for almost everything that we do. So like hearing, seeing, writing, reading, studying, anything that we do, making decisions. So some part of cortex is responsible for it, and then not that it's totally individually and separated, but we know that they're all connected to each other. So we have, as we know, a separated brain of right and left hemisphere, that we're not going to go into that, and then 5 main lobes of frontal lobe, which is mostly evolved and developed in humans for decision making, cognitive processing, and some of those main human characteristics. Our parietal lobe, which is responsible for knowing landmarks and locating things, and also learning and things like that. We have occipital lobe in the back of our head, which is responsible for our visual processing, and 2 temporal lobes in each side, which we'll go into a little bit more details. And then cerebellum down here for our motor actions and very already evolved activities. So there is also one other aspect of architectural in our brain that we know it as Brodmann areas today. So in 1909, Brodmann figured that we have different regions in our brain with different architectonics or cytoarchitecture, which basically means that we have each of these regions, that we have 52 of them. Each of these regions has its own, density or type of neurons that, it forms some sort of different, basically, texture that we know as, Brodmann areas, like, we refer to them as Brodmann area number, whatever, 12. So having that map helps us a lot today. Why? We know that, okay, so for example, we know that I'm going to study landmark recognition. And I know that apparently in the fMRI images, it's around, for example, this part of brain. So what region in our Brodmann areas does that, is that related to? So then, knowing that, we can today have maps like this that basically help us know which part of brain is responsible for what. So we know today that, change in Brodmann areas kind of corresponds with change in function. So that's very beneficial for us to know that we have a map now we can study and correlate. So I'm not going to go into details, but I'm just going to quickly mention some of these areas that we know today. For example, our visual area, as we said, is in our occipital lobe in the back of our head. The association area is on our temporal lobe for short-term memory, equilibrium, and our motor function is that yellow part that you can see, and then where is responsible for our speech muscles is number 4 there, and then our auditory area is, again, our temporal lobe here. And then emotional area is more in the middle of the brain, midbrain part that we said is more evolved or longer. And then our sensor association area is number 7, the orange part. And then olfactory area, number 8, they're little. And then we can see that, they are super different between, like, in the ratio wise between human and like rats. So yeah, I'm not going to go into the details of all of them. If you're interested, you can definitely look at them later. So we're going to mention a few different types of neurons or cells that help us in studying environment. So this one, what we can see here, what we're seeing here is studying one neuron, and these black dots are the spikes of the neuron. So this is called a grid cell. That what we're seeing here, that's one neuron that basically is in the entorhinal cortex of a rat, which entorhinal cortex in human brain is under the hippocampus, but this is a study of rat. So what this shows is that this is a simple empty environment, and the rat is navigating the environment. So there was nothing specific happening in that environment, but the cell was spiking in very specific pattern of like a grid, a hexagonal grid. So that's how Moser et al., they call this grid cell, that apparently this type of cell divides our environment into grids for us to navigate easier. The other type of cell is our is basically place and head direction cells. So there are 2 different cells. So place cells help us to understand where we are in the room. So again, spiking in specific places, when apparently what it does is maps our environment and tells us where we are. So which is super cool. And that exists in our hippocampus and other parts that I guess Thackeray can talk about them more if if you find some time between your time, because they are fascinating. And then we have our head direction cells in thalamus. So, which basically tells us which way we are heading, literally. So, and then we have our border boundary cells in, again, hippocampus, that tells us when we get close to a border of the environment that we are in. So, for example, if we're walking there, they are not spiking. If we get closer to a wall or a border boundary, then they'll start spiking. So, and hippocampus, this is again a rat study, but hippocampus in human brain locates there. And we also know that hippocampus is responsible for binding memories, so helping us navigate and also learning, remembering. So all of this help us to make some sort of map that we call a cognitive map. This started by basically that's the main concept in navigating the built environment and learning the environment, started by Edward Tolman. And so Tolman did the study of putting the rat here and the cheese here. So the rat was not able to see or smell the cheese, but it learned to navigate and eat the cheese. So that was the behaviorists era. So that's important to remember, that when behaviorists believe that if the rat learns where to go and eat the cheese, then they'll just learned process that for example, rat only turns right here. So if we locate the rat up there, the rat had learned to just turn right. So it should turn right according to behaviors. But the rat didn't turn right, so it turned left and ate the cheese. So basically that meant for Tolman that apparently the rat has this cognitive map, or the map of the area to, locate himself and then find a way to wherever he wants. So we're not going to go into a lot of detail. The cognitive map, as that for itself is a whole big field, and to study, because we had a conference of Crane last year. We talked about cognitive maps a lot, and finally we're like, so let's just say what cognitive map is not as of now, because it's being used in many different ways. So if you want to learn about cognitive maps more, definitely join Crane, join our next conference, and there is a lot going on with cognitive maps then.
So for behavioral data, as I mentioned, and like Simin also mentioned, how it plays roles in studying, using neuroscience for studying built environments. There are some impacts from built environment that can be captured through patterns of behavior, and then we can capture them with different methods. One common method is observation, and then there are two very generic technique in observation. One is direct observation, when the observer is looking at the event, happening right in front of their eyes, and then the indirect observation is remote and relying on observation of others or recording of the event. And then the observer can have three different roles. The observer can be completely participants in the study, and somehow blend into the studied environment, or can partially participate. And then this third role would be non-participant observer. And then combining the techniques and the role of the observer would be, there would be three situations for direct observation, and then obviously in indirect observation we just have non-participant observer. And when we should think of this method, each of these situation is when we want to have like an insider view of the event, and we want like to get completely blend to the study environment to even like understand the events happening from their perspective. We can use like the completely participant direct observation. In the partially participant, it is one of the most common one, and it's like when we want like an in-depth understanding of the social group, but still we want like to have an independent external view of the event. And then for the non-participants, it's useful when we want like to observe an event, but we want like to have to observe it from completely third view. And indirect non-participant observation is useful when we want like when it's not possible to have like direct observation regarding like safety or health issues or like regarding the time or geographic restriction. The other method is survey. So when we are using survey, there are like three assumptions and when we want like to choose survey. The first assumption, if we want to choose survey as our study method, is that the respondent can see and read in text format the survey. So it may not be suitable if the participants have visual, we have participants with visual impairments, older adult visual difficulties, or participants with literacy challenges. And the other assumption that we have when we want to use survey is that the respondent is both willing and able to provide rapid response. So it may not be suitable for like sensitive topics that participants are not comfortable to share, or like other situations, like the criminal behavior or other type of behaviors that the participants may not be willing to provide information, like truthful information. And then this third assumption is that if the participants must search for the information, they will go through all the like whatever records that they need to go through. So if you're like doing a survey for like let's say detailed information about participants' household energy consumption over like a long period of time, or the maintenance history of their house, maybe like this method would not be the best. But if we decided, considering all these three assumptions, we decided to use a survey, there are 5 format of questions that we can use in our questionnaire, and like based on the research design, we can select the research design and the research question. We can select each or combination of these things, and then there are references available if you want like if you want to know more about these methods and how like different question format impact the results. Interview is the other method we use for like collecting data. So there are like 2 types. One is like when we are interviewing one individual per session, and then the other is like when we are interviewing multiple people in one session, which is like focus. And then the first one is suitable when we want like in-depth personal insights, or when we have like interviewing for sensitive topics. And because it's easier to establish trust, and also like we will give the participants enough space to speak freely. But we should consider like it can be very time consuming, because like we need to interview each participant separately in separate sessions, and then if the topic is more related to like group dynamic, it might not be the best option. Focus group is good when we want like to have diverse perspective from multiple participants in one session. And also it could be useful and suitable when we want like to have brainstorming session on or to explore a group dynamic or group related topic, let's say like teamwork topics or collaboration related research question. And then it has its own things. So like they may lead to dominant voice, or like some participants may not feel comfortable enough, to share in front of others, or it may lead, like focus group may also lead to conformity. And then there are, again, different question types for interviews, unstructured, semi-structured, structured. And then the reason that like we are providing this overview is like just to have, an organized overview of like what are the methods, and then based on the research question, which one we could approach. And when we are like approaching these methods, we should like think of the ultimate goal of the research design, right? So we wanted like to capture all dimensions of a research question as much as possible, and then all diversity of our subjects as much as possible. So for interview question types, there are unstructured, semi-structured, and structures. And then based on the question type, they may be suitable. So for like unstructured, it's good when we want like to discover new theme. Because it's flexible, and like you have like one or two questions to lead the interview, but then the rest would be more of like following the flow of the conversation regarding the research question. And then the semi-structure is when you like develop a set of questions to guide the interview, but still you have, you allow for flexibility in the interview. And it's suitable when we want like to get deeper insight through probing, but still we have a framework. And for a structure is when we want like to gather quantifiable data, or when like there is limited access to skilled interviewers, because like for those two, we need to have skilled interviewers so they can like manage the interviews, and then they make sure that we are not going off topic and we're like following the research question. But structured interviews often used like in telephone interviews or like rating scale or multiple choices items is when we have the framework, but we want to gather quantifiable data, or like we want to have interviews in like with like more human resources that may not be like skilled in interview. And then there are some like things that we should consider for all the question types that we consider. And then the other method in collecting human behavior data is automated data collection. So there are three techniques that we can use. One is environmental sensors, and then these devices are passively capture information about the environment, but human behavior can be interpreted from this data. The nonverbal trackers are the devices that capture information from subjects, and then the verbal sensors they also capture information from subjects, but they're verbal sensors, right? So for like the environmental sensors is, for one example could be like CO2 sensors when we use for occupancy prediction or simulation. And for nonverbal trackers is like the under the mattress sleep trackers, we had a study at Centigrade using, this also a method, this method as well. And then the verbal sensor is like the motion trackers, step trackers that we can use, based on the research question. Okay, so, as I mentioned before, when we are like approaching our research question, we are thinking of, okay, how can I capture as much as possible, like different dimension of my research question? And there are some situations when we need like large amounts of data, and we need them in like extended period of time, but we don't have like access. Human resources. Also, when we want like to monitor dynamics, data is necessary to understand the behavior. And then
रोशन भैया का फोन आया था, वो बोल रहे थे कि तुम घर का
you need to tell them to mute themselves. Thank you. And then also it would be useful when we want to capture specific details that like movement patterns or like very detailed behaviors that like human observers may miss. But we should consider that they like when we are using automated data collection, we may not be able to capture like the context of the behavior, and then it can raise privacy issues, especially when it involves cameras and people may not feel comfortable to feel that they are being tracked even passively. And managing and analyzing this data requires computational power, specifically for the analysis part. And they have high initial cost, and they rely on a specific technology. And then the other things is that, like, because they are using this technology that have pre-programmed for whatever information that they're going to gather, there is a little flexibility for the researcher if any changes happen. And if like they want to modify their data collection or the type of the data that they're collecting. And then the last one in behavior data is digital trace. It's somehow the same as like the automated data tracking, but it's been useful when it could be useful when we are like interacting with a digital system, like online information system. It could include like using social medias or interaction with devices like smartphones or smart speakers. The next one is physiological data. As we discussed, this method has been commonly included in literature of like neuroscience for built environment studies, besides neural data. And there are some activities that can be captured through physiological data. There are two categories that included in physiological methods. One is biophysical, and then the other is biochemical. Biophysical is like when we use blood pressure monitor to check the blood pressure, right? And then the biochemical is when we use like laboratory tests for blood sugar. And then there are different data collection methods in physiological data collection. One is self-report, one is observation, laboratory test, and electronic monitoring. And then the example is like, let's say regular heart rates. It can be self-reported by the subject, by the patient, or it can be observed by the nurse, or it can be monitored electronically. So what we are going to like give an overview would be mainly biophysical and electronic monitoring. So there are like these are the ones that we are gonna review quickly, and we have like three measures here that measure, like there are cardiovascular measures, the ECG, PPG, and BP, and then we will discuss the skin measures, the GSR, and then the skin temperature, and then we will discuss EMG and eye tracking. Just okay. So for the ECG, PPG, and BP, so ECG is like using electrodes to monitor cardiac health, and it can provide accurate data regarding like heart rate and rhythm, and because it needs to have like electrodes to be placed on the body of the participants, it's may be not like a very natural or like comfortable, and it is mainly used in like healthcare field for like monitoring the heart health. Blood pressure is like the regular blood pressure that we know, and it has been used in built environment studies to mainly reflect on long-term impact of like a built environment on target population. Let's say older adults, they measure it like in one time, and then after like 6 months or 1 year, they measure it to see if like their specific features have any impact. And the most common in this category is PPG, which can like continuously monitor, the heart rate and blood oxygen level, and also changes in blood volume. And because it can be used in like a small wearable devices, it is like very common to be used in many studies in interaction with built environment. And all of when like we're considering, this method the same as all other methods, we have like to think of, okay, what are the things that we should consider? So for example, for PPG as one of the most common, we have to like know that there are little sensitive to motion, lighting, and even like the skin tone. So we should like consider these things when we are capturing the data. And for PPG, the based on how the wearable devices, sensors are designed, they can be placed on different, part of the body, and also like based on the type of the research question you have. So sometimes like you need your participants to interact with something or navigate through a space. So it depends on like what's your research question, and then what are the devices and sensors that you have. The galvanic skin response, GSR, and the skin temperature also commonly used in studying, human built environment interaction. They are both non-invasive, and they can like continuously collect data, and they can use be placed in like a small, can be used in a small wearable sensors, like a wristband and patches, and it's the same as like other methods that you should think of, like if they are providing a data, or if we can understand like the causality behind that data. So when we are using these sensors, any of this method, this is something that we should always consider, right? That this is symptoms that we are receiving from the participants, but like how we can interpret this data, and what do they mean, and what are the causality behind these symptoms that we are receiving. So for the GSR, it's suitable for collecting data on emotional arousal, like stress and excitement as well. And then skin temperature is also for like thermoregulation and emotional arousal. The skin temperature commonly used for like thermal comfort studies in built environment. One thing about like skin temperature that we should think is like that the changes in the skin temperature may be slow and may not always reflect, immediate physiological or emotional arousal. Yes, and then this is like the common measurement signs for both skin temperature and GSR. Can go to the next slide. Thank you. So EMG is also another method being used. It's used to collect data on like muscle activation. So since it can collect this data from muscle activation, muscle tension, and it can be useful for understanding how physical interaction with the built environments affect the body. It mostly be used for posture and movement studies, like physical activity, stress, muscle tension, physical engagement. And they can be affected by electrical noise and movement. You can see here, like the participants, they use it for a navigation study. Sometimes if they are not like placed correctly, they can pick up signals from, neighboring muscles. So there are things that we should consider when we are working with these sensors as well. So yeah, this is the facial EMG that's used for another study on like quantifying human experience in architectural space. So you can see here, the participants as well the sensors, and then again, like these methods are being used, in combination with other methods, and the whole point is like to understand, like as thorough as possible, the different dimension of the research question, and like variation of participants. And the last one is that we're gonna discuss in physiological data is eye tracking. Eye tracking is also like a non-invasive method, and it can provide real-time data on gaze direction, fixation, such as blink rate, pupil dilation, and it could be useful for like understanding participant visual attention and visual engagement with the built environment. And then we can like interpret data to like understand the focus, environmental distraction, and cognitive process. And since depending on the research question, it can have like a minimal interruption for the studied environment. And recently, like new eye tracking devices are integrated into VR headsets, so they are getting more frequently to be used. in built environment studies, because like you just like need to have the VR headset, and then the eye tracking devices are integrated with the devices. And the same as other methods, we like should consider that they are just capturing information from eyes, right? So if there is something that can be captured from those information, then it would be meaningful to use this, method and device. And the other things is that they can be affected by movement. It depends on like the device and how they have been designed. And sometimes look, because they have like where they might create some limitation for the like participants. So if we are having participants with sunglasses, eyeglasses, it might be a little intrusive for the experimental design. And here is like just an example of using eye tracking, and they also use like EEG and other method in like neural data that Simin will explain. This is like also for a location encoding, both for like the participants itself and also the location of other people in a space.
Hello again, everybody. So we're here with the neuroscience methods for studying brain and neurons. As Yasaman said earlier, we're not really going to cover all of them, but I'll mention some. So within our talks by Dr. Thackeray Brown, so far, we know that kind of we have a clue of what to study with the cells, like border cells, place cells, head direction cells, or what part of brains that we mentioned a little earlier. So, but how are we going to study that? So we have a few methods that they are typically used in neuroscience studies in general, but we're going to mention some of them here. So we've divided them with their strength. What type of strength do they have? Is it temporal? Is it spatial, which means that are they good in locating where we are studying, or are they good in basically telling us what was the time of the event and then getting us in that direction. And then we also have non-invasive and invasive methods, which basically means whether we are going to go into the brain or is it going to study the brain outside without really invading the brain. So this is the general chart that you have. It mentioned almost all, but not really all of them, but most of the neuroscience methods that we can study brain. So we have our vertical axis, which is the spatial resolution, which, as you can see, starts from synapses from molecule level, very little, from 0.1 micrometer to 1 meter. So from synapse level, which is the neural connections, the molecule level there, to the whole brain. So that's the spatial resolution. Like, for example, you can see that patch clamp recording is down here, which is in the level of synapses and axons, like our cell level. And then we also have this horizontal dimension, which is the temporal resolution. So, for example, when we are talking about fMRI, we're somewhere here between almost 1 second to 1 hour. So that's the time that we can record and we can basically get the information, the data from the brain. So we have, for example, lesion, which I will introduce later, which means damage to some part of brain. When we are studying that to get the notion of, okay, so for example, my hippocampus is damaged, what is it impacting? What can I do or what I can't do, and how does it impact my performance? So when we want to study that lesion, we have a wider time, for example, a year or a week or something like that. So that's in general how to read this chart. And then we're going to start with the single unit recording. So for single unit recording, you remember that beautiful neuron of us. So, and then with cell body, axon, dendrites, all of that. So what we do in single unit recording, we basically put an electrode either inside the neuron or around close to the neuron to read it. So if we put the electrode inside the neuron, this will be what we'll receive. So for example, what we'll see is a positive pattern because of, if you remember, the ions and the basically charge, the electrical charge that they had. And then that also means that if we put the electrode outside the neurons, that will be a reverse pattern that we'll see. So it will be a negative wave that we're gonna see. So it doesn't depending on what you want to study, you will make the decision. But if we study one either inside or outside, we can kind of get control of what the other one is like. So and then this, if you remember what Takri was talking about, the cells like the border cells or grid cells, these are this is how they are studied. So we put the electrode inside one cell in rat's brain, and then we'll see how it spikes, and then we'll get the heat map of it. We'll get a clue of, okay, so yeah, if this is a border cell, if the rat is walking here, then this starts spiking, then that's how we can make a decision or get the information of how this looks like. So this is the single unit recording. And then second, we're going to go to ERPs, which includes EEG and MEG. So ERP in general means event-related, stands for event-related potentials, which basically what we are recording here is the ions transmission, and what happens in that electrical changes there. I am not going to go into the details. That's not really my job, but we just will get a clue of what are we recording in EEG. So basically are studying when we are giving our participant an event, for example, I don't know, snapping or showing something or changing something, how their brain wave changes with regards to that event. So basically that's called event-related, potential or event-related capturing. So by brain waves, we mean these 5 type of waves. That the gamma is for heightened perception, learning, problem solving, cognitive processing, which is the basically the highest performance level. And then the beta is for awake, alert consciousness. The alpha is for physically and mentally relaxed. Theta is for creativity, insight, deep states, dreams, deep meditation. Delta is for deep dreamless sleep, loss of bodily elements. So, and we will see all of those type of waves in our EEG headset that we'll show later in our demo session. So, for example, we are showing this in to our participant. And then this is how we can get, okay, so for example, here we had a bigger wave or a smaller wave or something, some sort of change or nothing. That's how we can study whether, some event matters or not, or our brain is reacting to something or not. And then the event-related potentials, as we said before, are in EEG and MEG, which EEG is for basically electrical brain activity, and for MEG is the magnetic field that the activity is linked. So, and they have different tools, different machines, and they have experts to help us with that. And then we also have scalp ERP, which means, again, we will have the EEG or MEG as headsets or sensors on the scalp without, making any invasion. And then we have intracranial, which means going deep inside the brain, not deep, basically going into this. And then, So we are going to go over fMRI. Thackeray showed a few of some of the lab research that Omar is doing, and then we will later have Dr. Hugo Spears also talking about some fMRI studies. So basically what it does, it's fMRI means functional magnetic resonance imaging, which basically measures the blood oxygen level. So that means that, you know, wherever we have some sort of activity in some part of some region of our brain, whatever is happening there, we have more oxygen there because it's, you know, there's activity there. So I don't really know the physical, you know, stuff behind it, but what I know is that the level of oxygen level in our blood there increases. So that's how we can basically understand where is our the region of event. So yeah, that's me. And that's one of the studies that our lab member Viren was doing. That was a really cool study. It was about memory and music, basically the effect of music on our memory, if you're interested. So for fMRI studies, we have two main type of design, block design and again, event-related design. So for block design, as far as I know, we block the events into chunks, and then that will help us to better recognize the patterns. So for example, we are going to show blocks of, let's say, little houses, and then all of a sudden change the block into large houses, and then again changing into little houses, and then seeing the pattern or whether our attention or our brain activity shifts from somewhere to the other. So, and then for event-related design, it's more, I guess, timing matters more. It's more random. Am I right, Thackeray? Please, yeah. Okay, so that's how we basically can see the pattern. So depending on the activity, depending on what we want to see, what we want to study, we'll decide on which type to use. Again, so we're showing this, and then all of a sudden we're changing it into, basically, we reverse it, and then we'll see whether we'll see anything in fusiform, I don't know, face area. Why? Because this looks like a face or not. So that's how we can study fMRI. And then we have diffusion tensor imaging, or DTI. Again, we do this study with MRI machine, but this is a different type. That in this type of study, we basically track water diffusion in white matter, in brain white matter. So that will give us the connection. So what we're seeing, this beautiful artwork here, is our axons. So these are the connections, neural connections in our brain that we can see where they're connected to each other. So this also can help us to get the connectivity and, you know, which part of brain is connected to where, and, you know, where we have the wiring of brain. So, and then we have lesion that I mentioned a little earlier. So we have either a damage in the brain by an accident, an incident, something happened to someone's brain, or in a surgery, you know, intentionally a surgeon tries to remove some part of brain. So basically some part of brain is damaged. So that's how we can study whether this change in their brain or this damage right now in their brain changed any of their performance. So that's how we could get to know the importance of hippocampus in, you know, forming, binding memory, new memories. This is the most, I guess, famous psychology and neuroscience subject, HM's brain. So for HM, they removed the hippocampus basically in a surgery. And then later they realized that it can't form any new memories while he can totally remember a lot of things from his past, but he's not able to form any memories. So that's how we could learn about the importance of hippocampus. So far we talked about different methods. Yasaman mentioned some behavioral and some physiological data collection and methods, and then here we saw a few of neuroscience methods. But how do we know which one to pick? How do we know what is the good method for our study? So here we will see that it matters that what is our research question? What are we studying? And what is the research design? For example, is it between subject, within subject, or what part of the brain should we study? And that means that what part of brain, what is the scale? Do we need to focus on one cell, like what Thackeray was mentioning, place cell, border cell, some sort of cell that we need to study very, very in a very little scale? Or no, we want to study the entire brain and see what region of the brain matters. So do we have a region of interest or ROI? So if we have a literature on the topic, then we know that, so far we know that apparently hippocampus is responsible for some sort of memory shaping. So let's say if hippocampus also matters in associative memory or location memory or any type. So we have a region of interest that hippocampus does something for memory, but we are not 100% sure if that's the place that we need to study or not. Or we are kind of sure we want to see, what is the level or, what is the timing or anything like that. So that's how we know if we have a region of interest or not. We're just going to go more exploratory. And then whether our study needs to be spatial/functional, which again means spatial means that we have a space in brain that we know that region should be responsible for that activity, or whether there is any region, or whether that's only connections, or whether that part of brain does some sort of specific function, or we want to be more temporal. So for example, what is the reaction time for blah blah event? And then the other things that matters is that do we have expertise in the field? Do we have experts around us? Can we connect to the experts to help us? Or are we going to, what is the cost basically of this study? Am I going to make a whole career out of this and, just start learning, what EEG is or what fMRI is and, how am I going to run an fMRI machine and all that, which means that there's a lot of cost of, money and energy and all that. And then what is the noise or artifact level? So for example, we know that, yes, I know how to use EEG, but EEG is very noisy. And, if I'm going to wear a headset and, try to move around and, explore this environment and see the events, for example, if I'm passing this door, whether that changes or not, when I'm seeing a bigger isovist number of, for example, I have a bigger field now, or whether that changes anything in my brain or not. So we know that that's noisy. So we'll need some people to help us or, whether that works for us or not. And then the invasiveness that we talked about before. So can we, study the brain by an invasive method? Am I fine with that? Is my participant fine with that? Do I have that opportunity, or am I, collaborating with some people in the medical field who are already doing some studies or some surgeries that I can collaborate with or not? So what are the side effects? For example, we know that some people are not, okay with fMRI. They're like, yeah, so this is a magnetic field and I don't know what will happen to my brain. So that gives us some, again, limits. And then whether we are working on a specific disease. So for example, Alzheimer or any other type of specific things that we need to consider for the age of our participants. So, and then plus we also have some limits in our techniques that Yasaman mentioned some of them, and we can go deeper into some of the neurals methods, but we didn't cover them here. If you are interested, then we can definitely talk about that later, because we're definitely planning on, hopefully, if we have the energy and if we do our qualifying and dissertation and stuff, next workshops. So then we can, delve deeper into the neuroscience methods. So, for example, we know that for fMRI, some parts of our brain is not shown in MRI or fMRI images because in the temporal lobe, and they show as empty hollows. So, but we know that there exists some white matter and gray matter there that is responsible for language. But people who are interested in studying language, maybe MRI or fMRI is not the best way, so they study with PET or PET. So that's how we can kind of get to a decision of which method to pick. I know this is a lot, and I know we're like, okay, so then what am I gonna do? Which I should put all of that aside and not use them, but that gives up creativity. So that's where we know how to connect, and we make connections. We make basically we use our knowledge, people's knowledge, experts around us, Georgia Tech devices, or I don't know, collaborations and all of that. That is not disappointing. That is a great opportunity. We see it as great opportunity for all of us to study, from architecture to neuroscience to anything, engineering, anything in between that studies this. So we all can get together, help each other for making these studies moving forward.